Okay, so here are the light independent reactions of photosynthesis. Now, the aim of these reactions is to get carbon dioxide to turn it into carbohydrate, such as glucose, okay, C6H12O6. Now, right, what I've written down here is I've written the formula of some carbohydrates. This one is glucose, or it could be fructose. That's also got the same formula. This could be ribose, or it could be another one we're going to talk about. Look at in a minute, ribulose. And this one with 3 carbons in it is called glyceraldehyde. So we've got a 6 carbon, a 5 carbon, and a 3 carbon sugar. Now let's see what they've got in common with them. Well, if you look at the ratio, you can see that it's CN H2O N. Right. So for every carbon, you have 2 hydrogens and 1 oxygen. Now that's pretty important. Right. So now, before we have a think about this little formula here, I just want to talk about oxidation and reduction. Now, in organic chemistry, as we said before, oxidation is, you can think of it as adding oxygen or remove hydrogen. And reduction is the opposite of that. Remove hydrogen, so remove oxygen, add hydrogen. So whenever you do either of those, one's oxidation, one's reduction. Another thing to remember that oxidation is nearly always exothermic, whereas reduction is endo. So that's a lot harder to do, because we said endothermic reactions tend not to be thermodynamically possible. Most reactions you come across are all exothermic. So if you think of a molecule like carbon dioxide, that is very highly oxidized because you've got 2 oxygens for every 1 carbon, and you haven't got any hydrogens at all. If you have a molecule like methane, CH4, that's the carbon there is very highly reduced because it's got all hydrogens and no oxygens. If you have, on the other hand, let's think this one, which is called methanol or formaldehyde. That one is like a carbohydrate, really, isn't it? Because it's got 2 hydrogens for every 1 carbon and 1 oxygen for the carbon. Another one would be like methanoic acid. That would be like that. That is more oxidized than this one because you've got 2 oxygens now for the carbon, and so on. Anyway, that we'll have a think about that. Why am I talking about this when I'm talking about the reactions of photosynthesis? Well, the reason why I'm talking about it is because really what we've got to do is we've got to turn carbon dioxide, which is all the carbon there is highly oxidized, into carbohydrate, which is a lot more reduced. So there's got to be some kind of reduction going on there. And well, you may have come across reducing agents in biology, and if you've done glycolysis yet, you may have heard of NADH. Sorry, capitals, NADH. Well, in photosynthesis and in the light dependent reactions, you make a different reducing agent, very, very similar, which has got a phosphate attached, which is NADPH. Okay, so that reducing agent is very much involved in these reactions. Right. The reason why I've put down this structure here in the blue box, this is called glyceric acid. And if you have a look at this, let's see if this is a carbohydrate. We've got 3 carbons. We have got 3, 4, 5, 6 hydrogens, but we've got 4 oxygens. So you can see that it is more oxidized than a carbohydrate. So it's not a carbohydrate. Right. this is glyceric acid. If I knock off a hydrogen there, then we'd call this, it's just lost the hydrogen, behaves an acid. That's called, it's now called glycerate. And if I replace that OH group down there with a phosphate, an organic phosphate, so I'm going to call it P, that's PO4, 3 really. That is called 3-phosphoglycerate, and this is an important intermediate in these reactions, in these slightly independent reactions. But we can see here that it is not a carbohydrate, it's too oxidized, it's got too much oxygen in it, and if you want to turn it into a carbohydrate, you're going to have to reduce it, and you're going to need a reducing agent. Okay, so let's have a look at what happens in photosynthesis then, like in the reactions. Okay, now here we've got this sugar here called ribulose bisphosphate. Okay, you can see it's a 5-carbon sugar, and the only difference is it's got phosphates on the N2 carbon, so that should really be an OH group there, and you've got a phosphate group being stuck on there, and the same on the top carbon as well. It's got a phosphate there, but it's a 5-carbon sugar, and you can draw it simply like that, just 5 carbon atoms with a phosphate on each end. Now, this reaction here is catalyzed, so we're going to add carbon dioxide to this molecule, so plus CO2 here. And the name of this enzyme is called Rubisco. And I think I was reading that it is one of the most abundant enzymes on the planet, because every green plant has Rubisco in it to catch carbon dioxide, and there's a lot of plants everywhere. So anyway, what it does is carbon dioxide, it reacts to form with Rubisco, catalyzes this reaction to form this intermediate. Now that lasts for a fraction of a second. It's incredibly unstable. I've only just shown, you don't need to know the structure, but I've just shown you how the carbon dioxide sticks, you know, the green atoms there, these are the atoms of the carbon dioxide. That immediately falls apart to form 2 3-carbon units, and you can see that these two things here, they're the same as each other. One is just the upside, one is just the other one drawn upside down, and this is called 3-phosphoglycerate. This is the stuff I was talking about, which is not a carbohydrate because it's too oxidized. So in order to turn this into a carbohydrate, we've got to do some reduction. And it's not surprising it's too oxidized, because if you think you started off with ribulose bisphosphate, which is a carbohydrate, and then you add CO2 to it, and the carbon in CO2 has got a lot of oxygen, very oxidized, so obviously the products are going to be more oxidized than a carbohydrate. So the next steps are involved in reduction. Okay, so if we look at the reduction steps then. Right, here we've got this thing 3-phosphoglycerate. And what we want to do, we want to turn that into glyceraldehyde, which looks like this, which we mentioned before, which is a carbohydrate. So this is a carbohydrate. If you look at the formula, it's C3H6O3. This is glyceraldehyde. So we will have made carbohydrate if we can do a reduction here. So what have we got to do a reduction? We have got to, you can see we've got to replace, you've got to get rid of that O minus there, and we've got to put an H on it there. Now, you remember saying that reduction, so we're going to, well, reductions tend to be endothermic. Now you've got a reducing agent, we've got NADPH, and that in theory, because it could reduce it. NADPH, that's the oxidizing agent, it's reducing agent, sorry. That itself, it becomes reduced to that. That in theory might work, but it can't, because as that reaction would be pretty endothermic, and endothermic reactions cannot occur. So what the cell has to do is it has to use ATP to drive forward an endothermic reaction, because we remember the hydrolysis of ATP is very exothermic. So if you can do that, because this is so exothermic, it can compensate for the endothermic, the other reaction. So what happens is, before we do this, we've got to do it in a couple of stages then. Okay, so if I just move some of these things out of the way. I can go there. And push that there. So what you need to do is you need to react that with ATP first of all, and stick a phosphate onto this bit here. Okay, you need to stick a phosphate group onto there. So you need ATP. Where does ATP come from? It comes from the light-dependent reactions in the chloroplast. Of course, I should say that all of these reactions are occurring in the chloroplast now, but not in the stroma. Okay, so in the aqueous bit, not in the membrane, the hyaloid membranes. Right, so you've got ATP is going to come in here, and the last phosphate group gets transferred onto the end here. So we've got a phosphate on there now. Okay, so that would be called 1,3-phosphoglycerate, because you've got two of them. Got one carbon atom number 3, one and number 1. So we haven't oxidized it yet or reduced it yet, and in the next step we are going to reduce it. So the reducing agent the plant uses is NADPH. And so that gets oxidized itself, goes back to NAD+. And what happens there? Well, the phosphate remains stuck on the, I should put a phosphate on there, on that carbon number 3, that stays there. But you notice that this phosphate, right, the phosphate that was on carbon number 1, that's gone. Okay, so this comes off as an inorganic phosphate. And what you've got to understand is that by splitting that inorganic phosphate off there, that's very exothermic. And that compensates for this endothermic bit where you're reducing that to the hydrogen there. So you need ATP for the first step to kind of activate the molecule, if you like, and then you need to make the molecule unstable. I think I prefer to say that, and that means it's much easier for it to be reduced by the NADPH. So anyway, so we've got glyceraldehyde now, which is a 3-carbon sugar. 3-phosphate. It's a carbohydrate. And then what happens to it is some of that glyceraldehyde that can be converted into glucose and other sugars. Okay, but some of it has to be diverted to regenerate ribulose bisphosphate. Now, all of these reactions, because all you're doing when you're converting glyceraldehyde to glucose, you're not oxidizing or reducing, you're keeping hold of that carbohydrate ratio of, C1C to 2 H's and 1 O. So there's no, so all you're doing is rearranging. So those reactions, there's not much of, they're not exothermic or endothermic, they're all just generally rearrangements. The same here when you're making ribulose from glyceraldehyde, because they've got the same level of oxidation and reduction in the molecules, and so the energy changes aren't great. So it's just a matter of chopping bits of molecules together to form bigger ones. If you want to make glucose, that's quite simple because it's literally just 2 glyceraldehyde, 3 phosphates, and they can be stuck together. To make ribulose, well, you're going to need 5 glyceraldehyde, 3 phosphates, so that's 15 carbons, and that's going to end up giving you 3 ribulose bisphosphates. And there's a whole load of complicated intermediates involving 4 carbons, 3 carbons, 7 carbons, but there's no oxidation or reduction occurring in any of those reactions, and you don't need to know about them. They're generally speaking, they're not that interesting, really. It's just a matter of rearranging things together. Okay, so that is the light-dependent reactions. In this, we've only turned 1 CO2, incorporated that into a carbohydrate molecule. So, of course, to make 1 glucose, you'd have to have 6 carbon dioxides go through this whole process here. Okay.